Welcome to Biology My Passion. I am Soumya Harikrishna. We are learning the chapter Cell Cycle and Cell Division. Let us learn about the second type of division called the meiosis. All that cells in our body except the reproductive cells, that means we call them as somatic cells, they undergo division called the mitosis, where one mother cell will divide into two daughter cells with an equal number of chromosomes. So if mother cell has 46 pairs of if mother cell has 23 pairs of chromosomes, each daughter cells also will have 23 pairs of chromosomes. Now we are going to learn another type of division which is happening exclusively in the germ cells, or otherwise called the reproductive cells. In reproductive cells, this division is happening because the daughter cell will have only half the number of chromosomes. You know, if an organism is diploid, the diploid means what? Two sets of every chromosome will be there. That is, first chromosome 2 will be there, second chromosome 2 will be there, third chromosome 2 will be there, 21st chromosome 2, 22nd 2, 23rd 2. Like that, every single chromosome we will have 2. Why? One from father, one is mother. That is paternal and maternal copy of every single chromosome. That is why we call it as 23 pairs. That is the number in human beings. So any diploid organism will have the paternal and maternal copy of each chromosome, whatever be their number. So if the organism is diploid with the two copies of the same chromosome, when they undergo meiosis, they will have only one set. It's a mixture of these two. Don't think that only the paternal will come or maternal will come. It's a kind of mix. Okay. And the resultant cells are the gametes. Gametes are always haploid. So which cell can undergo meiosis? Only diploid cells will undergo meiosis. Can a haploid cell undergo meiosis? No. Because think of a situation, in our case, 23 pairs of 46. If it is becoming half during meiosis, it is only 23. That is haploid in our gametes. Can that 23 become half again, 11 and a half? It is impossible, right? So, only diploid cells can undergo meiosis. And when do they undergo meiosis? In order to produce the gametes or the sex cells. Now, the cells which undergo meiosis are called meiocytes. So, meiocytes are present in the sex organs like a testis in male and ovaries in female. And in this reduction in number happens, as I mentioned. Meiosis is a step which includes again substages. Prior to this, what should happen? The preparatory stage or the interphase stage. The steps are the same. DNA duplication happens during the S phase and the cell is ready for division. We are only focusing on the division phase, that is the karyokinesis here. Okay, so meiosis again divided into two phases, meiosis 1 and meiosis 2. Actually, during meiosis 1, the reduction in the chromosome number takes place, and followed by that meiosis 2 happens, which is similar to our mitosis, where the chromosome number is maintained. So meiosis 1 and meiosis 2, they both have again those four steps: PMAT, prophase, metaphase, anaphase and telophase. Here we call them as prophase 1, metaphase 1, anaphase 1, telophase 1. Suppose if I say telophase 1 means what? It is a telophase of meiosis 1. And meiosis 2, how will you say? Yes, prophase 2, metaphase 2, anaphase 2 and telophase 2. So let's start with the prophase of meiosis 1 or prophase 1. Let us dive into prophase 1. Prophase 1 is considered the longest phase of meiosis, and it includes 5 substages: leptonema or leptotin, zygonema or zygotine, pachynyma or pachytine, diplonema or diplotin, and diakinesis. So leptotin, zygotine, pachytine, diplotin and diakinesis. These are the 5 stages of the prophase 1. You can use an interesting mnemonics if you like. That is Lucy the zebra packed Dunkin' Donut. Okay. So here leptonema. Leptonema stage, what happens? The cell is there. Inside the nucleus is there. Inside the nucleus, chromatin fibers are there. They start to form the chromosomes with the 2 chromatids. And they are scattered throughout the cytoplasm. There is no order of their arrangement. So like this, they are scattered. Okay. So I will redraw this with the 2 different colors so that you will be able to understand. Now you can see total 2 pairs. In our case, how many will be there? 23 pairs will be there. Of this you can see 2 purple are same color I have drawn. They are the paternal, or the red ones are the maternal, or the other way around, whatever it is. But they are paternal and maternal. They are just scattered, and this stage is called a leptotin stage, where the chromatin material condensed to form the rod-shaped chromosome. Still the nuclear membrane, nucleolus all are present. Leptotin means it is the thin thread stage. Now, when we move on to the next stage, that is called a zygote or a zygodyma stage. In zygodyma stage, what happens? There I am drawing only the nucleus. So here, inside the nucleus, I am now not drawing nucleus also, it is there. Okay, so the pairing of the homologous chromosomes happen. Homologous chromosomes means corresponding chromosomes from father and mother, paternal and maternal copies of the same. For example, if this is first chromosome, this is the identical pair of it. The same way, if this is the second, this we can see from the size itself. That's why I drew it with the different sizes. So the paternal and maternal copies, they come together. This is called a pairing of the homologous chromosomes. So the other pair also, they come together. This is happening during the second stage, that is called a zygodyma. Zygo means what? Coming together or pairing. So in this case, what happens is, though I am drawing like this, they are coming and lying on top of the other like this. Suppose this is a chromosome. Imagine here also it is there. So another chromosome, this is paternal, this is maternal, they come and pair like one over the other. And once they come together, there will form something called a ladder-like structure connecting them. Okay, so one on top of the other, and there is a brush-like structure. You can see in this picture, ladder-like structure will form. It is made up of a protein, and that will connect them together. That is called a synaptonymal complex. And as a result, they are paired together. This pairing is called a synapse or a synapsis. When they come together, that synaptonymal complex join them. That is called a synapse, and the process is called a synapsis. Okay, once the synapsis is over, they are together. Now we move on to the next stage, pachynyma or pachytene stage. Pachytene stage is very crucial in bringing variation. Why? Because recombination happens during this. When they are held together with the synaptonymal complex, these two will exchange some parts between them. That means some part of the paternal copy will go to the maternal, and the maternal will go to the paternal. For example, if I draw like this, only one I will show you. This is the thread-like, actually the structure is like this, okay? And this other one, it is on top of it, but we cannot show that, so it is like this. Okay, so these two ends are overlapping. Now what happens is they exchange the parts. As a result, you can see here what will come, the maternal part. And here what will come, the paternal part. So exchange is happening. It is not only at the end, it can happen any place. So when this part is overlapping, so this part can come here, and here this will come. So this is called a crossing over. So crossing over happens during pachynyma. As a result, recombination or new combination of the genes are happening. Why? Because when they are coming together, each position here is a gene. Okay? Suppose for you to understand, if I number 1, 2, 3, 4, 5, 6, 7 like that genes, it's another form is here, right, in the maternal part. So 1, 2, 3, 4 corresponding gene from the mother also. So we call them as alleles. So alleles are there. So a few alleles of the father is exchanged with a few alleles of the mother's chromosome. Got it? So this is happening during the third stage, pachytene stage. Very, very important. Crossing over or the recombination happens during pachytene stage because of the lot of variations are coming in the child. Because we are, even though we are getting a paternal chromosome, it may have a few genes or alleles of the paternal part also. Understood? So it's a combination. Now coming to diplonyma stage. So once they exchange their parts, the purpose is met. Now they have to separate out. So now what happens? They will separate. Okay? Imagine before they were one on top of the other. Now they are coming to the side. So when they are separating also, suppose if I have my chromosome, other paired homologous chromosome, this maternal and paternal are called homologous. So as if I am holding my arm together. We are crisscrossing our arms. Okay, so now at this stage, even though we are separated, it's like this. Okay, though separated but still holding on to some part. So this also exchange already like this. Okay, so this is called, imagine we are crossing the arms. So this is called what? The chiasmata. So because X-shaped structure formed by this is called a chiasmata. Now we move further and further and further. So the hand will be held only at the end and finally we separate out. So that is called the terminalization of the chiasmata. So chiasmata will dissolve by the synaptonymal complex also will dissolve. Finally these two will separate out. That is called a diakinesis. When homologous chromosomes come together and lie on top of the other with the help of synaptonymal complex, it has two chromosomes in it, so we call it as bivalent. And how many chromatids are there? 4 chromatids, because 2 chromatids for each chromosome. Like that, 4 chromatids totally, so we also call it as tetrad. So in this stage it is more condensed, in this stage even more condensed. But remember, all these stages have nucleus in it, a nucleolus, but only at a diakinesis stage the nuclear membrane, nucleolus all will disappear. And also when synaptonymal complex is dissolving means what? They repel each other. So they came together, they fell on top of the other, they exchanged their materials, then only they started fighting. So the repulsion started. So they repelled or they separated and forming the X-shaped structure called a chiasmata. Then they separate along the chromatid. So that is called a terminalization of the chiasmata. Ultimately they two separate out. At this stage of diakinesis, spindle formation starts from the poles. Now we will move on to the next stage, metaphase 1. Followed by prophase, the metaphase starts. In metaphase 1, the homologous chromosomes will come and align at the equator. So that is the difference. Here, homologous pairs are arranged, like in the pair of 2, paternal, maternal, paternal, maternal, like that. But in the case of mitosis, how did we see? Suppose 4 are there, all the 4 used to align at the equator in a single line. But here, they are occurring as pairs. Now, during anaphase, what happens? This, meanwhile, the spindle fibers are attached here, like in the metaphase. Now, coming to anaphase 1, these homologous chromosomes will go to different poles. Okay, so like this it is pulled. Again, can you correlate the difference in the anaphase of mitosis? In anaphase of mitosis, what happened? They all were in the center, each one was split. Chromatids went to each pole. As a result, there suppose 4 chromosomes were, all 4 were split. 4 chromatids to one pole, 4 chromatids to the other pole. But here what happens? Homologous chromosomes are arranged. So exactly half number will go to this side and half number will go to that side. That means what? 23 and 23 will only go, not 23 pairs again. Okay, so here anaphase 1 is reducing the number to half by separating the homologous chromosomes. Okay, so in this case before it was 4, now each side only 2 are there, so the chromosome number became half. Now what happens? The telophase stage. Telophase stage means what? These two they rearrange and here nucleus will form. Okay, and this this is called a dyad stage. So the nucleus, 2 nuclei are there. This is called a dyad stage. At this stage, there can be cytokinesis happening or cytokinesis may not happen. So as a result, we can get either 2 daughter cells or we can get 1 daughter cell with a 2 nuclei. Now this will proceed to the next stage, that is meiosis 2. But before going to meiosis 2, what is happening here? A short break. The cell is telling, okay, I need to take a break. It is like our interphase. But it is not called interphase. Why? Because DNA duplication is not happening. Everything happened in the beginning. So we can call this short phase as interkinesis, not interphase. It's a kind of interphase, but DNA replication is absent. So it is called a interkinesis. Followed by interkinesis, these daughter cells will enter into the next stage of meiosis, that is meiosis 2, which is similar to our mitosis. So meiosis 2 is similar to mitosis because it is also an equational division where the number needs to be the same. Whereas meiosis 1 was reduction division. Understood? So again meiosis 2 has 4 stages. First prophase 2. In prophase 2 what happens? Sister chromatids are condensed and also the spindle fiber formation happens. But here this time the spindle fibers are formed right angles to those formed at the meiosis 1. So meiosis 1 if it was in this orientation, now it is from this side. Okay, and now in the that is the difference here. Then coming to metaphase, all the chromosomes will align at the equator. So from both sides the spindle will come and at the equator the chromosomes are aligned. Here again all the chromosomes are aligned at the equator. In that case 4 were there, all 4 will be aligned at the equator. Now we'll be coming and attaching, pulling it to both the sides, that is anaphase 2, where chromatids of these chromosomes are pulled to both the poles, and as a result what will happen, we will get 2 nuclei again. So total how many nuclei are there? 4 nuclei are there. Then cytokinesis happens, as a result 4 daughter cells are formed. So in case of mitosis we got only 2 daughter cells. Here after meiosis 1, 2 daughter cells called dyads, and then now we have 2 more, totally 4 daughter cells are formed. Hope you understand the concepts I teach very well. If so, please like, share and subscribe to my channel Biology My Passion. Thank you for watching.